Next one, protoplasm. Protoplasm of bacterium. It includes plasma membrane, cytoplasm, nucleoid, plasmid. Protoplasm of bacterium includes plasma membrane, cytoplasm, nucleoid. It also called genophore. And plasmid. Now first come to the plasma membrane. Inside the cell wall present plasma membrane. It is the cell wall and it is the plasma membrane. The structure and chemical composition of plasma membrane in bacterium quite similar to plasma membrane present in eukaryotic cell. That means the plasma membrane in bacterium also composed with lipids plus protein, lipoproteinaceous in nature. In some bacteria, the plasma membrane show infolding. Those infolding called mesosome. Infoldings of plasma membrane, mesosome. Function of mesosome. What function it performs? It is a prokaryotic cell. Except 70S type of ribosome, remaining cell organelles are absent. It compensates the function of those absent cell organelles mainly. Then what are the functions that perform this mesosome? 1. Cell wall formation. Cell division. Replication of DNA. Equal distribution of DNA into daughter cells. Secretion consists respiratory enzymes. Mesosome involved mainly in cell division. It involved in the formation of the cell wall. It helps in the replication of DNA. One DNA that becomes the 2 DNA. And this daughter DNA easily equally distributed into the daughter cell during the cell division. Secretion and consider respiratory enzymes. These are the functions carried out by mesosome. Here, mesosome is the infolding. When anywhere folding is present, area increases. So, mesosome also increase the area of absorption of nutrients. Mesosome increase the area of absorption of nutrients. Next, cytoplasm. In the protoplasm remaining one plasma membrane, the remaining substance that is called cytoplasm. In cytoplasm present only 70S type of ribosomes. All other membranous cell organelles are absent. Only 70S type of ribosomes present. In some photosynthetic bacteria, plasma membrane that invaginates and it separates from the plasma membrane consists the pigments called chromatophores. Chloroplasts are absent. In photosynthetic bacteria, chromatophores are formed. They are formed from the plasma membrane. Chromatophores are the only membranous structures in the cytoplasm with photosynthetic pigments. Chromatophores are originated from plasma membrane. Plasma membrane invaginates, consists the photosynthetic pigment, and it is separated from the plasma membrane. That invaginated and separated membranous structure from the plasma membrane with photosynthetic pigments called chromatophores involved in photosynthesis. Some bacteria consist gas vacuoles. Which bacteria consist the gas vacuoles? Mainly those bacteria, photosynthetic bacteria, grow in water, consist the gas vacuoles. Photosynthetic bacteria, photo means light, they require light. Gas vacuoles are impermeable to water but permeable to air. When they are filled with air, they help in the float of the bacterium on the surface of water or nearer to the surface of water. So, gas vacuole helps to bacterium to float on the surface of water or nearer to the surface of water for getting sunlight. So, gas vacuoles present in aquatic photosynthetic bacteria only. Example, green. Photosynthetic bacteria. Purple. Photosynthetic bacteria. Green photosynthetic bacteria, purple photosynthetic bacteria. They are aquatic and photosynthetic. For getting sunlight, they must be float on the surface or near to the surface of water. Gas vacuole helps to them to float on the surface of water or nearer to the surface of water. Reserved food materials present in bacterium. Reserved food materials present in bacterium also called inclusion bodies. Reserved food materials present in bacterium or the cytoplasm of bacterium, inclusion bodies. The reserved food materials are, in bacterium, the reserved food materials are glycogen granules or phosphate granules. They are also called volutin granules. PHB. It is the abbreviated form of PHB. Poly, beta, hydroxy, butyrate. Reserved food material in cytoplasm called inclusion bodies. They may be glycogen granules or phosphate granules. Phosphate granules also called volutin granules, and another one poly beta hydroxy butyrate. It is a fat reserved substance. Phosphate granules or volutin granules, they are the source of phosphate for ATP synthesis. Glycogen is the carbohydrate. It is a polysaccharide. Glycogen is a homopolysaccharide. Homopolysaccharide of glucose molecules. Glycogen. All these are the reserved food material in cytoplasm. Next one, nucleoid. The main genetic material of bacterium called nucleoid. Main genetic material of bacterium nucleoid. Also called genophore. Bacterium is a prokaryote. True nucleus is absent. Consists, the genetic material consists single naked chromosome or naked DNA arranged in circular manner. Histone proteins are absent. So, DNA associated with non-histone proteins only. Nucleoid is double stranded naked DNA arranged in circular manner. All the genes present in the nucleoid are responsible for to perform various metabolic activities or life process. In some bacteria, in addition to this nucleoid are the main genetic material present, small, circular, naked, double-stranded DNA, plasmid. Plasmid not present in all bacteria, in some bacteria. Along with this nucleoid present small circular double-stranded DNA, that is naked DNA, that is plasmid. Plasmid may or may not present. Genes present in the plasmid not perform any metabolic activity. So, without plasmid, bacterium can survive. If we separate this plasmid from the bacterium, no harm to the bacterium, it can survive. Reason, no metabolic activity performing genes are absent on the plasmid. These plasmids may present freely in the cytoplasm or it may attach it to the nucleoid. If plasmid attach it to the nucleoid like this one, it is the nucleoid. If the plasmid is attached, it is the plasmid and it is the nucleoid. That is called episome. Plasmid which was attached to the nucleoid called episome. We observe such type of episomes in HFR strains. HFR strains. Episome observed in HFR strains. In normal strains, plasmid that present freely or separately, nucleoid and plasmid they are present separately. Genes present in plasmid not perform any metabolic activities. Then what function they perform? Some plasmids, the genes secrete toxins and enzymes. In some plasmids, the genes are involved in reproduction. In some bacteria, the genes present in the plasmid show resistance to antibiotics. Based on the function they perform, plasmids are three types. Based on the function they perform, plasmids are three types. F plasmid, R plasmid, Col plasmids. Based on the function they perform, plasmids are three types: F plasmid, R plasmid, COL, Col plasmid. Now first come to the F plasmid, also called F factor or fertility factor, involved in reproduction. Involved in reproduction. That we will discuss in detail in conjugation in the sexual reproduction of the bacteria. R plasmid, R here, resistance. Genes present in the plasmid show resistance to one or more than one antibiotics. So, such type of plasmid, they show resistance to antibiotics called R plasmids or resistant plasmids. Col plasmids, these are the plasmids, they secrete toxic substances. Full form, carcinogenic plasmids. Col plasmid means carcinogenic plasmids. They secrete toxins. One of the example to this toxin, bacteriocin. These toxins kill other harmful enterobacteria. So, carcinogenic plasmids secrete toxins, they kill or harmful to other enterobacteria present inside the organisms. So, these are the different types of plasmids, only 3 types of plasmids based on the function they perform. Genes perform metabolic activities completely absent in the plasmid. So, in biotechnology, we remove the plasmid. In such condition, bacterium can survive. We introduce the plasmid into the vector or the host bacterium. In such condition, also bacterium can survive. No effect to the metabolic activity, either presence or absence of the plasmid.